Hello, can anybody hear me? Give me a thumbs up. Maybe. Thank you. Okay, you can put your name in the chat. That'll let me know that you are here. Thank you. I'll wait a little bit and then I'm going to review for the exam. Final exam is this week, opens Friday and will close Monday night of next week. So I think it's the 14th through the 17th, if I remember. So no lab tomorrow. We're just getting ready for the final. I have to stall for a minute. So we'll see. Got a minute. Okay. Let's see. About half people are here. All right. Sorry, I forgot. I needed a link today, so maybe some people might be late because I just had to send a new link. So we'll see how that goes. All right. Okay. I have grades to put in, so I'll be putting grades in tonight and tomorrow. So we'll see that. Should be mostly caught up after tomorrow, but you never know. All right, well, let me, it's almost time, so let me start my sharing up. And so here we go. You should see, let's see. Does anybody see Albert Einstein with his tongue out and a thing that says review? Give me a thumbs up, thank you. Okay, so their final review is mostly comprehensive. So probably about 2/3 of it. So it'll cover things like natural selection, evolution, talk a lot about the evolution of the animals and the animals as a group, but also describe the other major groups of organisms and the life cycle and in their kind of importance. That will be our focus for that. The final exam is worth 150 points. That's like 1.5 of the regular exams. That it'll have, oops, there's a short answer there in diagram. There won't be any short answers in diagrams. There will be the same 2 essay questions. There will be multiple choice and there will be some true false. There won't be fill in the blanks. There won't be short answer in diagrams. So I left that in there. It'll cover primarily evolution and then a real exploration of biodiversity with some emphasis on animals and probably plants secondary to animals. But there'll also be stuff about bacteria, archaea, and fungi. So you know, that's our focus. The bacteria, fungi, and the viruses, that there'll maybe be a little more emphasis, particularly bacteria and viruses. You know, okay. So, you know, the textbook, there's not time to read it anymore, but there's time to look at the chapter summaries. You can look at the glossaries for each chapter, what's in there, kind of the index, the review questions, that kind of stuff. That's at the end of the chapters that we covered. And like I said, there'll be some emphasis on those last 2 chapters, the prokaryotes and the viruses. You know, you have your, on Moodle, you also will have the study guides for each chapter. Those are good resources because they give you kind of a summary of important points and a list of keywords. And so those are good resources to use. And so you can't reread the chapters, but you can probably and should reread all the chapter summaries, look at those terms from each chapter. Ones that look unfamiliar, then go look at those study guides. That's stuff that you could do for all of them. And, you know, in some of my classes, I talk a lot about how to study. I do it less in this class, but like in in my lower division classes. We want to put things in long-term memory. In long-term memory, we know how that forms in people's brains, that forms because of repetition, and it forms because you've been working with something. So doing active things like saying the words, writing the words, using flashcards, stuff like that. So use active methods like making notes, making review sheets, quizzing with a partner. Those are much more effective. And we know repetition works. So that if there's repetition, it's better. And it's better to use a bunch of small sessions, 15 minutes, 3 times a night, and 5 nights out of 7 nights. That's really helpful. We know long-term memory forms during sleep, and so it's good. It's good to get a lot of sleep the week before finals and before final to get sleep. Those would be really helpful things. I'm hopeful that you guys know that. And again, this is science. And so we're trying to kind of talk about some scientific things. And, we're talking about, when we talk about phylogeny, this is all, these are all based on evidence. What is the evidence? So it's testable, an idea that we're trying to match with existing evidence and come up with new evidence. And so that's really the key thing. It's not a conspiracy theory. It's what is the evidence. And so that's something that we're always trying to do, is talk about evidence. And when we talk about evolution, then that's kind of the big idea that is behind the course of biodiversity evolution, is the idea that there can be genetic changes in a population of organisms. And these are changes that occur because the frequency of some genes can increase and the frequency of others can decrease. And of course, new genes show up because of mutation. And depending on whether those are good in that environment, those genes may become more common. And so this is what's led to new adaptations and new species. And when we're talking about evolution and biodiversity, we're talking about the really long picture. So we're talking about fossils. What are the simplest groups? What are the oldest groups? Which groups are most different? We're looking at transitional forms. And so in natural selection, it's just the idea that some individuals have genes that make them more likely to reproduce and survive. And so that leads to genetic change. That's the main mechanism. Now there's those other mechanisms like genetic drift, mutation, sexual selection, non-random mating, but the main driver is natural selection. So all those other mechanisms cause change, but natural selection over long periods of time will be the big one. Okay, and so when we talk about evolution, that some individuals are more fit. And that doesn't mean, when we say fit, it doesn't imply that they're bigger or stronger or more fierce or faster. It means they're better at leaving offspring. Okay, and so if you're a slug that's really, really slow, but nothing can eat you, and you can find all the food you want, and you can produce lots of offspring, that's better off than being a T-Rex, because if a T-Rex needs all sorts of food, and it has small populations, and so being a slug might be much better. And like these two guys here, the one guy spends all his time in the gym and injecting steroids, and that shrinks his testicles and lowers his sperm count. And the other guy, you can see he's got 2 kids. That's higher evolutionary fitness. And so that's the whole point, is evolution works on leaving more offspring. So genetic change in populations means that there's got to be variation. The variation has to be transmitted through genes, and that they have some differences in their relative success for reproduction. Okay, so the Hardy-Weinberg principle we talked about was allele frequencies. don't change if there's random mating. It's a large population. There's no migration, no mutation, and no selection. So you can say evolution will occur if there's non-random mating. Evolution occurs in small populations by chance. New populations, it occurs when there's migration. It occurs when there's mutation. And then, of course, when there's natural selection. Okay, so we can talk about evolution in a micro sense, where it's within one population, but we also talk about it in a macro sense, where we're talking about it in longer time scales, where we're talking about big geography pictures, where there's species and adaptations. Both of these things apply, and we can see them both, and there's evidence for both. Okay, and so the evidence for long-term things that we use for evolution is that we can see this with artificial selection. We've been able to produce different varieties of dogs. We've been able to produce different varieties of corn and wheat. We've got cows that produce tons and tons of milk. These are all signs that there's lots of variation, and that the variation is genetic, and depending on conditions, that some variants will do better than others. We also see lots of fossils with transitional intermediates, different sequences of fossils. We see older fossils have simpler organisms, and that gradually organisms got more complex. So there's all these things. And for the last one is that biogeography, that's for Darwin and Wallace, that was a big thing. Why were islands had so many unique species, and why did, what explained that? Well, because they got there and they changed. And so that was one of the best pieces of evidence. So there's fossil record. Archaeopteryx, Archaeopteryx, a really good example of a transitional form, because it's a fossil intermediate, because it clearly had feathers, but it's got features no bird ever had, or no living bird has. That it's got claws and fingers on the wing, it has teeth in the mouth, it has a bony tail, and its rib cage and hips are not fused. But otherwise, it's clearly looking like a bird that could fly, but probably not as good as a modern bird. So it's a fossil intermediate. Homology is the idea that you have structural similarities that are due to shared common ancestors, so descent. So like the bone structure that you find in the leg of a person, or the arm of a person, the foreleg of a cat, the bat's wing, the porpoise, the horse, these are all formed in similar ways. So it's the same tissues, same embryonic tissues are active, same genes are active, and that's why you have similar patterns of bony elements. Evolution works to produce livable organisms. It doesn't necessarily produce the best structure. So like if you want to have a long neck, it would be best to have lots of vertebrae, but for some reason in mammals, the vertebrae are limited in number. All mammals have only 7. So giraffes, which have a very long neck, they can't do what dinosaurs or geese do, where they just have a large number of vertebrae in there. They just have huge cervical vertebrae that are elongate, and so they make them very, very big. Not as flexible as the neck of a goose or a dinosaur, but it works. Likewise, the eyes of vertebrates, the photoreceptors face backwards. And so here you have on the right side the eye of, I think that's a mollusk. And on the other side, you have the eye of a vertebrate. Light's got to pass through the nerve cells. So you have a blind spot, and you're not going to capture all the light. But the squid eye there on the other side works. It maybe works slightly better, but vertebrates have a lot of things better than squids do. They have, they have a better system for swimming, and so squid get large and they do okay, but there's more fish, and of course, some fish and some mammals eat squid, even the largest squid. So it's not so much the eye, it's other things. Okay, and then things like vestigial structures. Okay, evolution explains things like the hip bones in whales, which never get on land. No, they have no visible external legs. Structures in humans that don't appear unfunctional. This is a boa constrictor. They have these little nubs where their back legs should be. And there's lots more like this. Okay, and then evolution also predicts that certain forms should reappear, that because selection pressures might be similar. And so for four-legged animals that climb trees, it might be good to be able to jump really, really far. And so you have a bunch of animals that have developed a membrane between their forelegs and their hind legs so that they can glide. And so essentially they can jump really, really far. And so you have flying squirrels and phalangers, or sometimes we call them sugar gliders. They have very similar lifestyle, but they've independently evolved that flap of skin. And this talked about new species. How do species happen? Species is a group of interbreeding populations. How do new species develop? Well, they have to become isolated and then they change. And usually this is due to geography, but it can be due to other things. They could be behaviorally isolated, particularly if there's barriers to mating. That's really important. And once organisms have changed enough that they can no longer produce fully fertile offspring, then you've kind of got a new species going. So that reproductive isolation. Okay, and so we can know how evolution produces new forms, new species. We've seen it happen. And so one way that we think we see this happen is with what are called adaptive radiations, where a close number of species seem to descend from a common ancestor. And often this happens when they get to a new habitat, or they have a key adaptation, or when other organisms go extinct. So that might be like the honey creepers. Key innovation is where they develop a new structure, like a new beak or something like that. Or in the case of fish, the ability to have a swim bladder that then becomes connected with the airway. And so they can burp up air or swallow air. That led to development of a lung. And so you had fish produce all these organisms that used lungs. Okay. Also with rapid change, the idea that there's mass extinction and that you have periods where there's this punctuated equilibrium. And we can see this, that sometimes the fossil record changes abruptly, like at the end of the dinosaurs, and then suddenly you get new species that occur after that. So it becomes quite different. Let's see. This is what is this? Okay, this is from Derived and Ancestral Characters. This is when we're talking about classification of organisms. We look at new features, those derived characters, organisms that share new features. That's kind of cool. And so then we produce them using the principle of parsimony, the idea that the fewest number of changes, the fewest number of branches is the best idea. And so a good example of this is grouping birds with some of the dinosaurs and the crocodiles. Otherwise, you'd have to assume that things like hollow bones might have evolved twice, that parental care might have evolved twice, air sacs evolved twice, the four-chamber heart evolved twice in different, or 3 times or 4 times in different groups. But here we say that it's probably that they're evolved once, and that the groups that show more similarity have gone extinct. And then when we look at feathers, we can see this even more clearly. And then we can look at things like hollow bones, wishbones, and things like that. Those things are there. Okay, so just goes through that examples, parental care, stuff like that. Yeah, for me it was when I realized that alligators and crocodiles have parental care. They lay their eggs in a nest and they don't sit on them, but they watch them. That was a big thing because go down to Florida, I thought maybe we'll see this. And so if you ever see baby alligators, run, because the mother's probably not far away. Okay, so let's skip over this. Biological species, interbreeding populations. Let's go. Okay, so here, what's in here? Okay, here's the virus stuff. Bacteria and viruses evolve really fast. Evolution occurs fast when reproduction rates are high and mutation rates are high. Here's phylogeny of the animals. So here, you should be able to give me some of this stuff. So when we talk about evolution, we talked about animals in a lot of detail. And so here, this has more detail than you should give me. But the idea is that the animals are clearly related to single-celled eukaryotes, some sort of protista, probably flagellates, because most animals produce flagella and have flagellated sperm, so their gametophyte type stage, their haploid stage, is flagellated, and so that strongly suggests that they come from flagellates. And probably the simplest animals are the sponges, and they have flagellated sperm, and they have flagella in the diploid state as the sponge. Okay, and then and they lack symmetry. And then the first group after that is two distinct body layers, true tissues, true muscles, and those are probably the radially symmetrical organisms like Cnidaria. Those are the jellyfish and corals. And then you have the bilaterally symmetrical animals that split into two major groups, the protostomes and the deuterostomes. They all have 3 layers of tissues, endoderm, ectoderm, and mesoderm. They all form a blastula, which is a hollow ball of cells after so many cell divisions. And then they all form a gut by having an opening kind of appear through there by having migrating cells. The protostomes, that first opening that appears is the mouth. In the deuterostomes, it becomes the anus. But otherwise, they seem pretty similar. And then we break it down into the Lofotrochozoa, which have genetic similarities. They share maybe some larval stages and a feeding structure, the lophophore, some of them. And then the Ectisoa, which share a cuticle that they produce that produces like an exoskeleton. Those are all protostomes. And then the deuterostomes, that's the echinoderms and the chordates and their relatives, which we talked about. So with this, we're looking at anatomical features like symmetry, body cavities, and germ layers, to a lesser degree segmentation. So symmetry is, how can you cut them so that you can produce 2 equal sides? In bilaterally symmetrical animals, it's only one way. Okay, and then the other key features have to do with early development. So how do they produce cleavage, or what happens to the fertilized egg? How does it go through mitosis? So most protozoans have spiral cleavage, and it's very determined. So by the time you're at 4-cell state, each cell has kind of decided, I'm going to give rise to these body parts. That happens much later in deuterostomes. Okay, and then how they form the mouth and the anus, and how they form the third tissue layer with the mesoderm and the coelom. That's slightly different in these groups, and so this slide goes over it nicely. Okay, here, this is the Lofotrochozoa. The lophophorates have a feeding structure that's got tentacles that have cilia and mucus that trap particles. and the other organisms form a trochophore larvae, which is a little larvae that has bands of cilia to swim. Okay. So this includes like the cephalopods, the annelids, things like that. Cephalopods are my favorite because they're large and active predators with big brains and just really strange. So I like them that way. And then the other major group of protostomes are the Ectisoa. These have the cuticle, and that cuticle contains chitin, and they have to, it forms an external skeleton which has to be shed when they grow. And unlike most other animals, these guys have amoeboid sperm, and so they get rid of the flagellated sperm. And the deuterostomes, the echinoderms, as adults, they have 5-part radial symmetry, but the larvae have bilateral symmetry. They have this unique water vascular system and an endoskeleton, and they're related to the chordates. And the chordates have this body plan with 4 features: the pharyngeal gills, notochord, dorsal tube, tubular nerve cord, and postanal tail. Okay, the vertebrates, they replace the notochord with vertebrae eventually. They develop a cartilage or bone skeleton. They start developing a really well-developed head and organs, and they become a large group of large dominant animals. Okay, the primates are the group that's closest to us of these. These are mammals, so they are fur. They reproduce typically of mammals. They retain their young, their amniotic egg with the young, and they have grasping fingers, binocular vision, very mobile upper limbs for climbing trees. We're part of the hominoid groups, which includes the apes. They have grasping hands with opposable thumbs. They have fingernails, not claws, no external tail. Their tail is internal. Large brain, adaptable behavior, and slow reproduction. So they live a long time. Okay, and here fossil intermediates are like one of the best ones is the Neanderthal. Has lots of human features, but just slightly different. Was a contemporary of modern humans for a while in the Middle East and in Europe. Large brain, actually larger than modern humans, bipedal, used stone tools, made fire, made shelters, made artwork, stuff like that. Okay, so then we can do the same thing with the plants, okay, that we do with the animals. All plants are related. We know why, because they share gene sequences that have been taken from the ribosome. They show their chloroplasts all show the same similarity with cyanobacteria. Here we have the simple plants look a lot like algae. These include things like liverworts, the hornworts, stuff like that. The mosses, again, still have a dominant haploid organism, or most of a long-lived haploid stage that might be the dominant stage, like with some of these guys here. But then we wind up with organisms that have vascular tissues. So the vascular tissues, the tracheophytes, they start with that. And then the ferns have really good vascular tissues. They start producing roots when you have that. You start having your tissues become more specialized. And then the seed plants become even more specialized. They have more full developed vascular tissues. And then what's their really big advantage is that instead of having flagella that have to swim from one plant to the next, their flagella, or their haploid reproductive cells form a pollen in an egg and are transferred by either the wind or by insects. And that turns out to be good because they can explore dry environments. Okay, and so with plants, you have this life cycle. And we have this with mammals too, where there's a haploid and a diploid stage. But in the non-tracheophyte plants, the haploid stage, the gametophyte, is the dominant stage. and it's the sporophyte that's relatively short-lived. And because the fertilization is by a haploid sperm that swims, these organisms are tied to really moist environments. So an example of one of these non-vascular plants would be like a moss. Okay, and there's lots of species and they're all over, but they never get much larger than, like, an inch or two high. They're only found in damp environments. The gametophyte is the dominant stage. The sporophyte grows from the gametophyte and is temporary. It's much shorter. And they don't have true roots. They just have rhizoids that are holding them down. They tolerate drying. They don't really transport oxygen or water. They have to, for them to be active, it has to be really moist. Okay. So it's ferns that are the big advance. Ferns have true roots. They have vascular tissues. They are able to get much larger. They still have a gametophyte, but it's smaller. And the gametophyte is like only temporary. It's small. It still does photosynthesis, but it's temporary, and the sporophyte grows from it. We're almost to the point of more advanced plants, which happen like when we get to the gymnosperms. The gymnosperms produce true pollen. They produce true seeds. The seeds can be dormant for years. And so they can spread over dry environments, and they can just wait it out till they get water and the right kind of, right conditions to become active. All the gymnosperms are wind pollinated, so they tend to produce small windborne pollen. They usually will have male cones and female cones, and they will develop the seed right kind of in this modified branch-like structure. And out of the gymnosperms, it's the conifers that are the most dominant group, trees and shrubs. In some places, conifers are the best plants because they have these needle-like leaves, so they can do photosynthesis in very, very dry environments, and very, very good plants. Okay. Then when we get to the angiosperms, it's a little bit different. Here we have a double fertilization occurs where again, it's pollen. So you have this very small microgametophyte, and the pollen again has to land on the female part of the plant, but then one of the pollens will grow into a tube, and that will deliver the flagellated sperm to the oocyte and produce the seed, and then the tissues of the ovary will produce a fruit around there. Angiosperms, these are the flowering and fruiting plants. So you have these modified leaves for reproduction, and these are the relatively new plants, but they're by far the most successful. And so we have good fossil records of them, and they're, maybe 200 million years old and dominant after the Cretaceous extinction. So that's kind of a fruit production there. And then this is the dicots and the monocot. Here's a dicot that refers to the leaf structures that form in the seed and then have this arrested development. And so that gives you the flower structure there, modified leaves and things like that, stamen, carpel. And then there shows the cotyledons. There's also the monocotyledon plants, so they only form one leaf. And then you see the root meristem there and the structures of that. And there shows fruit. It's got the seed and the fruit. So the part of the seed is the endosperm, and the endosperm is the stored energy that comes from the polar nuclei and the sperm. So there's a double fertilization there. And then the fruit develops from ovary tissues around the seed. Okay, so there's a fruit. There's drupes, different types of fruits. Okay, yeah, we saw that. Okay, so then a big thing recently is the prokaryotic cells. There's two different groups, the archaea and the bacteria. They look almost alike, but they're genetically as different or more different than plants and animals. So the bacteria include aerobic and anaerobic organisms. The archaea include a bunch of different organisms. All of them are anaerobic. These include methanogens, the hypothermophiles, the halophiles, sulfur bacteria, things like that. It appears that the archaea share some similarities with eukaryotes, some of them have linear chromosomes with multiple units or multiple origins of replication. So there gives you a prokaryotic cell. Okay. Yeah. So they all look alike. They all have a cell wall, different materials in archaea and bacteria, but they all have a general shape and appearance that's similar and similar organization. Your book made kind of a big deal of gene flow and genetic types of recombination. So, this was stuff kind of talked about in Biology 1, so I didn't spend a lot of time on it, but it's there in that chapter. And then they talked about in prokaryotic cells, they have these extra ways of mixing the genes. So we talked about a little bit in evolution about sexual reproduction and sexual selection. Bacteria don't have that because they're always haploid, but they can exchange gene by having these gene transfer mechanisms, where living cells can acquire new genes. Sometimes they just pick up DNA, that's transformation. Sometimes viruses will inject DNA that they can use, that's transduction. And in conjugation, they form a little tube which DNA can transfer across between living cells. Okay. So this is useful. We adopt this, but this is kind of Biology 1 stuff. But when we're talking about prokaryotes, we should recognize that they're really diverse metabolically, that they can do photosynthesis without oxygen, without producing oxygen. And so they can be in ponds where oxygen never forms and never is there. They can use sunlight in some of the sulfur bacteria or non-sulfur bacteria to help eat other compounds. And then they can do things like oxidize iron. They can oxidize forms of nitrogen, all sorts of crazy stuff. And then we talked about a little bit about different pathogens, bacterial pathogens. And so they mentioned TB. This is just a thing on TB. It's was a huge problem in developing Europe and the United States. It was a very common chronic disease where the bacteria are multiplying within tubercles. And so that's why it's tuberculosis. And so if weakened hosts, this would happen. And so if the hosts were under stress, you could get this, but the infection could go latent. Okay, and that there's also, we should mention, lots of beneficial bacteria. They do important nutrient cycling. We use them to make food, to do food fermentations, to make cheese, yogurt, chocolate, beer, and wine. We also have them in our intestines, and they do good things for us, and we use them for industry. Then lastly, we wound up with the viruses. These are, some people will say they're not organisms, but they do a lot of the things that organisms do. And certainly when we're infected, we feel like there's an organism inside us. So these are intracellular parasites that are very chemical in nature. They can even be crystallized. They're much smaller than cells, and they can have genomes that are single-stranded or double-stranded, that are DNA or RNA. They can just do things that cells don't do. In general, there's a genome. That genome is small, very short, sometimes as few as 10 or 11 genes. That it could be RNA or DNA, single-stranded or double-stranded. They have a protein coat around the nucleic acid. That could be It could form a helical structure, or it could form a polyhedron, or sometimes even a complex structure. And then they may have part of the host membrane forming an envelope around it. Okay, lots of different types of viruses. They do lots of interesting things. Some of these are very, very important. This is human immunodeficiency virus. All viruses attach to cells. All viruses have to enter cells, or at least their nucleic acids have to enter cells. And then once they're inside, there's this replication and assembly of the virus, and then the virus has to exit. The exact details differ depending on whether the virus is made of RNA or DNA, whether it's double-stranded or single-stranded, stuff like that. Okay, prions are another kind of infectious, non-cellular organism type thing. Prions are protein only, and they replicate by converting normal protein to form the abnormal shape. So these, as a group, form the transmissible spongiform encephalopathies, mad cow disease, Kuru, Creutzfeldt-Jakob, Scrapie. These are all these diseases. And so these are brain proteins that convert to the abnormal fold. Oh, fungi, I forgot about fungi. Eukaryotic, multicellular, single or multicellular. They're all heterotrophs, no photosynthesis, and they secrete enzymes though, rather than ingest particles. They contain chitin, poor fossil record. Okay. They all have a filamentous growth form, often in soil and through materials, and they can often withstand harsher conditions than plants or animals. So dry conditions, acids, they can withstand that. Their cells may or may not have septae. They may have more than one nucleus per cell sometimes, but unique that way. Usually when we see them, they're this mycelium. It's a mass of fungi. It kind of acts like a single-celled organism, but eventually it'll reproduce and produce the mushroom form, which is the true multicellular structure. In liquids, these can form yeasts, which are kind of rounded cells. Okay, I think that's it. You don't have to know that. Okay. Okay. All right. Does anybody have any questions? That was long. Yes? No? Okay. Okay. So the exam opens Friday, and on Friday you can take it then, or you can take it Saturday or Sunday or Monday, but it will close Monday night at 11:55. Okay, you have 2 hours. Okay, I will post essay questions. I didn't have them ready yet, but I will post them probably later today. So it'll be same as the usual, 2 essay questions, maybe worth a little bit more, mostly multiple choice, mostly true and the false. Okay. Will they be different from other essay questions? Yeah, they'll be new. There'll be at least one on viruses and one on bacteria. So there'll be some new stuff there, and you'll probably have to figure that you'll have to answer at least one of those on viruses or bacteria. So, but there'll be one on, I think, one on evolution. What's the other? There's one on evolution, probably one on animals, phylogeny, and then it's bacteria and viruses. So it's a good question. Anything else? Okay. Let me look back, see anything else. That's all I see. Could we go over the essays on a review session on Wednesday? Nah, I think I could probably do that. Let me get a pen and write it down. Is 2 o'clock good? Yeah, okay, let's do that. 2 o'clock on Tuesday, I'll post the essay questions before then. Okay. Okay. Anything else? All right. So Tuesday you can, or you may come. Otherwise, I'll just post it and you can work there. If I have a chance, I will get some practice questions available for us as well. Okay. No, there's no lab tomorrow, so you can use that time to start reviewing. I will post some more grades later tonight. I have the worksheets corrected, so I'll post all those. And let's see. Okay. But so Wednesday at 2, we'll talk about the essay questions. Okay. All right. Okay. All right. So I will watch the announcement section for a new link for Wednesday. The PowerPoint that I showed you is available on this week's Moodle box, and in that same box, I'll post the questions. And I'll try and keep it short when we meet on Wednesday. All right, I think that's good. So we'll say goodbye and study hard. Again, you have until Monday before you have to take the final exam, and I will post grades probably by Wednesday. I'll have that done. All right. Okay, we'll see you. Bye.